Hello, hi. Today I would like to share or present regarding on DNA identification of cyanobacterial toxins gene and identification of the species producing the toxin. We are in a group 1. Me, myself, Muhammad Faisal Nisa, and my friend Fangiano, Zhu Meishuang, and Zhu Pei. DNA profiling, or we call environmental DNA profiling or DNA fingerprint, which is an important technique for analysis of diversity and characteristics of environmental microbes such as toxicity and metabolism of bacteria. The understanding microbe characteristics in an environment is important for various reasons, including early detection of harmful bacteria and possible change of climate or environmental conditions. DNA profiling involves 5 processes. First is DNA extractions, second, polymerase chain reaction (PCR), third, electrophoresis, fourth, bioroid gel dock SR imaging system, and lastly, sequencing. DNA extraction process. DNA extraction is a fundamental technique used in molecular biology to isolate DNA from cells. It involves 5 steps. First, lysis, followed by binding, washing, drying, and elution. For our labs exercise, we use bacterial DNA extraction kits. These kits also involve all the buffers, including proteinase K. First step is the lysis process. First, we are taking the sample from Toxin Harapan USM. Second, we must labeling our tube samples by write down the name and also the date. Third, we adding 400 microliter water samples of the Toxin Harapan to the tube by using pipettes. And next steps, we centrifuge at 6000 for 2 minutes. This process is a mechanical technique, which is centrifugal force is used to separate components of mixtures based on their size or density. And followed by the next steps by adding 100 microliter of buffer R1s, and 6 steps adding 20 microliter of lysozyme to the tube. The lysozymes is function for makes the outer cells crush or breaking to release of DNA from the nucleus. First step of these steps is to make the outer cell crush. 7. Incubate at 37 Celsius for 20 minutes in a shaking water bath. At the incubation process is to ensure a complete lysis of cells in the suspensions. These digest proteins, suspends lipids, and also digest or break down cellulite materials and make DNA free. And again, we centrifuge 10,000 at 10,000 for 3 minutes, and followed by adding 180 microliter of buffer R2 to the tubes. And for the next steps, we add 20 microliter of proteinase K into the tube. Proteinase K is to make the cells break down. It helps to make the cells break down. Continue to the next step. We incubate at 65 degrees Celsius for 20 minutes in a shaking water bath. And next, we add 440 microliter of buffer BG to the tubes. The buffer BG is function to make the pH stable, the same pH, the pH of our sample in stables. And followed by incubate at 35 degrees Celsius for 10 minutes. That's the ending of the lysis process. And next is the binding process. For the binding process, we add 200 microliter of absolute ethanol. absolute ethanol is functioned to make the DNA bigger. And then we transfer to the sample 650 microliters into the column assembles, which we can see at the center of the tube just have a filtrations. And then followed by the centrifuge at 10,000 for 1 minute, and at 17 steps we remove the debris at the bottoms. We can see at the bottom is a debris here, and the upper top is a DNA stuck there, because the DNA become bigger by using the absolute ethanol. And in this process we repeat for the remaining samples from the step 14 until our samples finish, and we got the DNA stuck at the upper top of the filter. Next process is washing, drying, and DNA elution process. We use wash buffer by adding 650 microliters of wash buffer and centrifuge at 10,000 for 1 minute. And then for the column drying process, we centrifuge 10,000 for 1 minute to remove residual ethanol. Followed by the next steps, DNA elution by adding hundreds microliters of elution buffer and stand for 2 minutes. And then centrifuge 10,000 for 1 minute to elute DNA. And final, DNA will settle down at the bottoms for the DNA bacteria of the DNA bacteria that we get. And don't forget to name and write down the dates for our samples. And for the analysis we use the PCR. This is the pictures of the PCR process. For the left features, we can see the samples by adding the components: TAQ polymerase, primer 227F, primer 1492R, H2O autoclave, DNA templates. And then we put the samples into the PCR machines, and we fill out, or we setting the system according to the method here, the temperatures, the time, by follow these instructions to the system. And next steps, we analyze data by using the electrophoresis, by use the agarose, and then you put the samples at the instruments. Here you can see the samples, and we run, and at the end we get the results from the electrophoresis. This is the BioGel Doc SR imaging system, which is the gel documentation system, also known as a gel doc or gel imagers. I used to record and analyze the result of the gel electrophoresis and membrane bulleting experiment. These instruments are necessary for visualizing, stain or label nucleic acid and proteins in media such as agarose, acrylamide or cellulose. And lastly, analyzing sequencing. So this is the sequence that we got from our results. For group 1, we need to analyze 2 patterns of sequence. For the sequence ones, we are using the NIH BLAST, National Center for Biotechnology Informations, by putting the sequence into this website. And what we found from this sequencing, the bacteria that we detect at the most highest 99.35% is Cynobacterial species LKSZ1 DNA. The accession number AP031572.1. And for second sequencing, we are detect the Pseudana baina CF Galeta legi ribosomal RNA gene at the percentage 98.72%. The accession number MF629808.1. So we can see when we're running this sequence in this website, we will get the highest number of the species in the DNA bacteria. Here we can know what type of bacteria in our samples, the highest here for the first sequence and the second sequence. Okay, that's all. Thank you.